Hello, everybody. Thank you for being here. It's very exciting to see so many people interested in genetics at a software conference. That's definitely very unusual, but very cool. So genetics is a very cool field for a software engineer to be in. My name is Miro Cupak. I spend my days at DNA Stack, which is a Canadian company where we're building a cloud platform for genomics. And I spend my nights with a nonprofit organization that's called the Global Alliance for Genomics and Health, which is basically a standards body in genetics. It's a coalition of over 500 institutions at this point, institutions from research, life sciences, IT, healthcare, disease advocacy, you name it. Basically, organizations working together on standardizing how genetic data is stored, processed, and most importantly, shared. So I'm here today to talk about a particular project that I've been working on under the umbrella of this organization, and it's sort of a pioneering effort in genetic data sharing. The project we're talking about is called the Beacon Network. And it's an open source project that's very dear to me, something that I'm very passionate about, because I really do believe that data sharing, or lack thereof, is one of the most pressing issues in genetics right now. Genomics is a comparative science. When you want to analyze your data, you typically want to take your data and look at it in the context of ideally as many other samples as you can. And a very common problem that researchers face is actually just not having enough data. And it's not necessarily that the data doesn't exist. Sometimes, quite often actually, the data has been collected, it just hasn't been made accessible. So data sharing really is the key to advancing biomedicine and ultimately improving healthcare. The Beacon Network is one of the first successful initiatives with this focus, at least at a global scale. The project started about 4 years ago as my Google Summer of Code project, actually. And then got some traction, became my master's thesis, and now I'm actually fortunate enough to still be working on this. So I'm definitely very biased, so much for the disclaimer. So why are we talking about this, besides the fact that I enjoy speaking about this? Well, to our knowledge, this is the largest search and discovery engine of human genetic mutations in the world, which is pretty cool. And when I say it like this, the largest search engine, you're probably thinking that the kind of issues that we've been dealing with are issues around performance or scalability. And I would be lying if I said that we never hit any of those issues. We did. But the biggest issue for us was actually standardization. Like I said, GA4GH is a standards body. And from the very beginning, this was developed as kind of a driving force behind the standard and basically an application on top of a standard. So just getting the right people together, getting them to agree on things, and developing this as a standard, that was the really hard part for us. But I think the fact that this is in genomics, that it's one of the more successful data sharing initiatives there, and then that it was developed as a volunteering effort on a global scale is such an interdisciplinary environment, I think all these things make it a pretty interesting case study. So let's start by taking a look at the system. And I will try to do a live demo here if my connection is OK. So if you go to beacon-network.org, you will see a screen like this. That's sort of the main deployment of the service. There's a couple of things going on here. There's a search field. There's a list of participating organizations, some press coverage, links to documentation, etc. But the cool part is really this search field here. And we call this a search engine here, and you can search through this application. But maybe a better term would be discovery engine, because the discovery part, discovering where the mutations are, is actually the main use case for this service. So in this search field, I can specify the genetic mutation. And I'm just going to select the really well-known one here. It's something that has been associated with the BRCA2 gene, which has been linked to breast cancer. So I know that I'm going to get a lot of hits in the system. So we specify the genetic mutation in this very special format. It's actually not that special. If you're in genetics, you would recognize the format from other tools. But if you're not in genetics, it's still pretty easy to explain. As I'm sure most of you know from high school biology, you can imagine DNA as a string of letters, a very long string of letters, about 3 billion of those per person. And there's only so many letters in the alphabet of this string, or bases. And you might remember letters A, C, G, T as sort of the main bases featuring here. So to specify a genetic mutation, I need to say where in this long string it is and what the actual change of the letters is. And that's what I'm specifying here. Now, because DNA is such a long string, we typically divide it into smaller, more manageable chunks, like chromosomes or genes. And this brings us to the query. So the first number here is actually a chromosome identifier. And humans have 23 pairs of chromosomes, so this would be number 1 to 22, or X or Y. And there's actually a few more options because there's things like mitochondrial DNA, but let's not get into these details. The second number here is a position within the region, within the chromosome. So this would be a number between 1 and, let's say, 260 million, depending on the chromosome. Different chromosomes have different sizes. So this gives us the location. Now we need to specify the actual letter. And because I've given the exact position, technically all I need to do is specify that one letter or a group of letters that's in that position. In this case, I'm actually specifying a diff. So in this case, I'm saying a healthy individual would have on chromosome 13, position 32 million something, a letter G. But I'm looking for an individual that has a letter C there. And there are a couple of reasons why this is specified as a diff. Basically, it allows us to better capture more complicated mutations. For example, when a really large string gets inserted to a certain position, which would effectively offset everything else. But it's also a very common optimization. You know, we're all humans. We're all of the same species. So out of the 3 billion letters, the vast majority of them would be the same for all of us. So in practice, quite often, you don't actually need to store all of them. You might just want to store a difference over some sort of a reference genome. And that's what's happening here. So the first toggle at the beginning is actually the reference genome identifier. So I've specified the mutation. I'm going to click search. And what happens here is the search engine takes the query, it processes it, it talks to a bunch of services, a bunch of resources of genetic information on the web, and it aggregates the responses. So there's quite a lot of data going on here. We have a lot of organizations, really large research institutions like Wellcome Sanger Institute or the Broad Institute, as well as large data aggregators such as Google. And for some of them, they only tell you, OK, I have seen this mutation before, and if you want to know more, you need to contact me. In some cases, they actually show more metadata, like, for example, here, and that gets sort of nicely parsed. And sometimes they just tell you, OK, here's some information, but if you want to know more, you need to go to this external system, which typically would be behind an authentication and authorization server. So the important thing to note here is that we don't impose any constraints on how the data is accessed. Each of these organizations is completely in charge of what they expose. So sometimes the data might be completely public. Sometimes you might need to log into a system. Maybe you need to be verified as a bona fide researcher. Sometimes if it's really sensitive data, you might need to go through like a full scientific and ethics review by a data access committee. The point is that it really depends, and it's really up to the individual institutions. Our approach is basically show you as much information as we can, and ideally either link you to an external system or give you some contact information where you can find out more. And that's pretty much all there is to it. There's a few more things that you can do with the system, but this really is the meat and potatoes. So going back to my slides, you can see that the Beacon network is basically a federated aggregator, right? So why is such a thing even needed? Well, this is what the world looks like right now. The cost of genome sequencing has been going down dramatically. It decreased over 1 million times in the last few years. You can now obtain genetic information for a person for less than $1,000, and machines capable of doing this for less than $100 are expected in the next few years. So a drastic decrease in sequencing cost. And maybe unsurprisingly, this led to an explosion of genetic data. The total amount of sequenced data is doubling approximately every 7 months, beating the Moore's law. So if the growth continues at the current rate, we're expecting that we reach 1 exabase of sequence per year in the next few years, and 1 zetabase in the next 10 years. So in practice, this means that we're looking at tens of millions of genomes from large sequencing projects in the next 2 years or so, and we're looking at about 100 million up to 2 billion people sequenced in the next 10 years. So that's a huge increase. What does it mean? Well, we have a couple of problems, right? We have too much data for any single institution to handle, basically. So the traditional approach of aggregating everything in a single centralized site, which is how things have been done until now, it basically doesn't work anymore. And there's a lot of fragmentation in that aspect as well. The data is siloed. It's often split by a disease or by an institution, by a country. And it cannot really be shared easily because there's regulations in place or just incompatibility of methods and tools. Secondly, we still don't have enough data to make new discoveries. We need more. And the key obstacle is really finding additional patients, discovering the new affected families. So it's pretty clear that the federated system capable of executing queries across datasets, across institutions, that's the missing piece here. And that's exactly what the Beacon network is trying to pioneer. So the Beacon network is called the Beacon network because it connects all these services. And these services individually are called beacons. And this term comes from a GA4GH initiative that was called the Beacon Project. It's something that started back in 2014. Basically, GA4GH recognized that data sharing is the key and data sharing is needed, but they didn't know if the world was ready to share data. So the premise for the Beacon project was basically to just test willingness of organizations to share. And this came in the form of a proposal for this simple web service that you can implement, and it exposes some information about your database of genetic data. So the proposal goes like this. Create a simple web service that accepts a question of the form, do you have any information about this mutation? And it responds with yes or no. So it's really very simple. There's literally one bit of information that it exposes. And that was kind of the goal. And later, once people bought into this, we actually extended this. You can now expose things like allele frequency or number of matches or arbitrary key value pairs. But the simplicity in the initial design was absolutely crucial to the adoption of the project. And there were actually 3 design principles that kind of supported simplicity from several different angles. First of all, Beacon had to be technically simple. Remember, this was an experiment, right? If it was expensive to implement or it would require too much work, nobody would participate in this experiment. And we're dealing with research institutions here, right? Not software shops. So technically simple means actually really, really simple. A Beacon also had to minimize risks associated with genetic data sharing. So the idea was to expose so little information, that one bit, that nobody would think that it violates anyone's privacy. And this actually turned out not to be the case. It turned out that that one bit is actually still too much for certain things, but that's a conversation for another time. And it also had to be possible to make beacons publicly available. Public is obviously the most open form of data sharing, which is very good, but it also means that you don't need to deal with things like authentication or authorization, which makes it even easier to implement. So, like I said, GA4GH was a standards body. So in the last 4 years, this went basically from an idea to a pretty mature standard. So let's briefly take a look at this journey. When we first introduced the Beacon network, which was back in 2014, there was basically no formal API specification. All we had was this one-sentence description. This is the question, and this is the answer. And you might think, it's such a simple API. Surely everybody is going to implement it well. Well, it turned out that wasn't the case. When we introduced the Beacon network, there were 4 beacons at the time, and they had completely different APIs. One of them was using JSON, one of them was using XML, then we had plain text and HTML. They were using different request methods, completely different parameters, different notations, different domains for the same parameters. It was just a mess. Also, when you created a new beacon, there was no sort of automated process of registration. You basically had to email GA4GH and tell them, hey, I have this beacon here, can you add it to your website? And somebody would manually go and add this sort of free-form text to the website. So each beacon would have a section like this, where they would describe how a particular beacon is queried, and so on and so forth. So it basically didn't scale, and as a consumer of this API, you were in a terrible situation. If you wanted to find out if anybody has seen your genetic mutation, you would have to go to this page, read it all, understand it, and then write a script that actually talks to all these services, deals with all the API nuances, and somehow extracts some meaningful information from this. And that's actually how the Beacon network started. It was just a bunch of converters between all these different APIs, accessible from a single place on the internet. So Beacon network unified the API to some extent, which led to the first version of the standard, version 0.1 at the end of 2014. The first specification was really simple. What you're seeing on the right is actually not an extract. That's the whole specification. So there was a very simple Boolean response, just ran false. It was written in Apache Avro, which is an open source data serialization system. Basically, it comes with this interface definition language, Avro IDL, where you specify your schemas and protocols, and then you can compile them to a data model in various programming languages. So it's pretty good for this sort of thing, where you need platform independence and such. The specification certainly was a step in the right direction, but it shared many of the disadvantages of the original Beacon definition. Specifically, it was too vague. We basically defined the data model, but we didn't say how it maps to the endpoints. We didn't say what kind of output format you should use. We didn't clarify the domain of the individual parameters. So it was still really vague, and that was the problem in adoption. So we thought, OK, fair enough. We learned the lesson. So a year later, we came up with version 0.2. Now, we added a few interesting features to the API. We added support for data sets as means of organizing our data, data use conditions, so you could say things like, this data can only be used for research, or in this country, which is quite common with this kind of data. We also added new response types. Aside from true and false, you could now say overlap and null, which allows us to better capture insertions and deletions. In retrospect, this actually turned out to be a very bad design decision, because not many people are actually dealing with insertions and deletions. And it turned out that the reason people were buying into this was the simplicity. And they really liked the fact that they could say that this is a service that responds yes or no. So now that this was no longer true, that was actually a surprisingly big obstacle. Maybe more importantly, we were painfully aware of the vagueness of the previous version of the specification being kind of the reason why this didn't get adopted. So we tried to compensate for that, and in a way, we went overboard and tried to overprescribe everything. So it actually led to a very complex protocol, which in turn also caused problems in adoption. An interesting fact is that around 60 beacons were created during the course of 0.2, but strictly speaking, only one of them was compatible with the spec. So everybody else decided to diverge from the specification for various pragmatic reasons, which as an API designer, this tells you that you basically developed a standard that doesn't match real needs at all, which is a problem. So we thought, fine, lesson learned, let's try again, year after that. And with this release, we really took a step back, and we basically decided to break backwards compatibility in favor of creating really nice, stable API as a basis for potential future stable release. We got the right people involved, and we really focused on simplifying everything. We focused on modularity and extensibility so that we can evolve the protocol later on. And we focused on development of supporting tooling. So this was the first time that we had things like a reference implementation or a compliance suite, SDKs, CLIs, various data adapters for various data sources. So we basically tried to make it really easy for people to create new beacons that would be compatible with the specification. And we actually really got it right. This was a very successful release and very positive adoption. We also decided to switch away from Avro. We hit some of its limitations, and we went to protocol buffers. So now, earlier this year, we came up with 0.4, and we added a few things, like support for more complex mutations. But basically, we just focused on polishing existing things to really just bring us closer to the stable API. We switched to the third format, OpenAPI, because basically nobody was happy with Avro or protocol buffers. People find it hard to work with. And so far, we've been super happy. It was really the right call. And finally, this brings us to 1.0. So during this year, 0.4 was promoted through a series of patch releases to 1.0, which is now ready in the sense that it's on GitHub. But it's actually in that final phase of the official GA4GH standards review and approval process. So it hasn't been approved yet, but it is something that we're hoping to announce next week, actually, in Basel, Switzerland, which is the annual GA4GH plenary meeting. So we're really close. And it's a really exciting milestone for genetic data sharing, and really just the culmination of the effort of so many people over so many years. So really excited about that. So that's the standard. Let's talk about the search engine built on top of it. This is what the system looks like. Structurally, it's a collection of 9 subsystems, 18 modules total. It's pretty standard technology stack, old school if anything. It's using Java EE, now Jakarta EE, of course. JBoss technology stack, Wildfly, etc. It's running as a collection of services in Docker and Kubernetes. What isn't nowadays? So let's take a look at the individual subsystems and see how this fits together and what they do. So the data subsystem is actually pretty easy to explain. It basically takes care of data access. In our case, this is a relational database, MySQL, access for JPA. The service subsystem is sort of the core of the whole system. It takes care of communication between all the other components, and it takes care of things like query normalization. Basically, we support several different notations and several different conventions as inputs, things that people are used to from other tools, but internally we convert everything into a single canonical representation. The service also deals with aggregators. We have these concepts of virtual beacons, which are basically they're basically virtual services that from the outside they behave as individual beacons. They give you the right response. But internally, they're actually proxying to a group of beacons. So when they receive a query, they distribute it to all these beacons and then aggregate in a single response. So in practice, basically, you end up with this directed acyclic graph of beacons because it's many-to-many relationship. Parents can have multiple children, and children can have multiple parents. So this brings us to participant resolution. Whenever the service receives a query, it basically performs a breadth-first search on the directed graph, and it comes up with the minimal list of services that you need to talk to be able to answer a query. So it accounts for things like aggregators and caching, and gives you like the bare minimum of network calls that you need to execute. So this is actually quite a significant optimization for us, and it really helped us get the response times down. It also takes care of query distribution. This is basically level 1 parallelization in the system, kind of across all the beacons. So service parallelizes across beacons. Processor takes care of execution of a query against a single beacon. And it does that by running this 4-stage execution pipeline. Basically, the problem that we had was that there were so many differences between different APIs that static configuration wasn't cutting it anymore. We needed something more dynamic, something more easily extensible, something more flexible. So we were looking for a way to store business logic in the database in some way. And this is what we came up with. So it's this 4-stage pipeline that's crafted individually for each beacon whenever a query is received, depending on the query. And the 4 stages are represented by a converter, a requester, a fetcher, and a parser. These are implemented essentially as arbitrary Java classes, so there's maximum flexibility. There's just Java code. Each of these components has an interface. So there would be a converter interface, a fetcher interface, and so on. All the implementations would implement that interface. They're kind of like functional interfaces in Java, but not really. They're basically functional interfaces with support for generics and support for Java EE. So they would be implemented as CDI beans or HABs, depending on what's needed. So when a processor receives a query, it basically reaches into the database, fetches the definition of the pipeline, then resolves the classes, dynamically loads them, compiles the pipeline, and runs it. So this is done in case of CDI beans. This is lookup by the class name, and then use CDI bean manager to instantiate it. In the case of EJBs, this is a simple JNDI lookup. So this is sort of level 2 parallelization. Sometimes you might need multiple queries against a single beacon to answer a single query in the beacon network. So let's take a look at the execution pipeline. The first stage is the converter. The converter basically takes care of translating parameters from the internal canonical representation to whatever language a particular beacon speaks. So there are 5 types of these for the main 5 parameters in the query. And just to give you a sense of like the scale of differences that we have here, we have 15 converter implementations as CDI beans. The second stage is the requester. The requester takes the parameters from the converter, it takes the URL, and it constructs a request that needs to be sent to a particular beacon. And we have 11 requesters, again, implemented as CDI beans. The first stage is the fetcher. So the fetcher is the unit that actually talks to a beacon, the unit that talks to an external service. So it takes the request from the requester, it executes a network call, deals with networking errors, etc., and obtains the raw response. And we have 2 implementations of these, both over HTTP as EJBs. So once the raw response is received, it goes to the parser, which is the final stage in the pipeline. And parsers basically take care of extracting information of interest from the response. So they deal with things like various data formats, JSON, XML, string, HTML. They deal with different fields in the response. They normalize the values. They deal with parsing errors, that sort of thing. So this is sort of level 3 parallelization. And we have 18 parsers implemented as EJBs again. There's also a thin mapper layer, which basically takes care of translating between different representations of objects. And there's also a very thin REST layer, which is basically API gateway. So it's the part of the system that interfaces with the outside world and deals with the client requests. We have 2 APIs, legacy XML and JSON. And aside from supporting all the functionality of the network, we also wrap each individual beacon in like a compatible, full-featured, unified beacon API. So you can actually submit standard beacon queries against the network. And this is actually quite challenging, especially when it comes to supporting very first beacons that didn't conform to any specification. And of course, it gets more and more challenging as the API is evolving. But so far, we've been successful at kind of backward supporting any beacon that has ever been created. So that's where we are. So we have this search engine, right? And it goes beyond any single data set, single beacon, single institution. So it's kind of in a unique position in that it has a global view of what people are searching for. So it would be kind of a shame not to do some data science around it. And I'm not a data scientist, but I did spend some time crunching some numbers. And I was basically looking at the last year and a half of usage data, which is since the last time we've done this kind of a study. And this is what I found out. In terms of the size of the network, we currently have over 100 beacons from 40 different institutions across 18 countries and all the continents, besides Antarctica, obviously not a lot of genetic research going on there. And because of the way this is set up, it's actually really hard to estimate how much data we're searching, because it's up to each individual institution to control that, right? So quite often they don't tell you the size of their data set. If I had to extrapolate from the information that we do have, I would estimate that a full search touches over 2 million samples and over 2 billion genetic variants, where variants would be unique within a data set, but obviously you have no way of comparing uniqueness across different data sets. So that's a lot of data, and I think we can truly call this a global system. In terms of the actual user base, so far we've had 16,000 unique users from 140 countries. So again, pretty global system. Maybe unsurprisingly, most traffic comes from the countries that also host beacons. That would be the case of 8 countries in the top 10 list. As for the number of searches, in its 4 years of existence, the system has served over 1 million searches. We actually reached that almost exactly a year ago. So, and that was a big milestone for data sharing, so I like sharing that number. They're sort of steadily increasing traffic, so right now we're actually closer to 1.5 million, but 1 million is a nice number to share. And since you typically target multiple beacons within a single query, this actually resulted into close to 8 million queries being distributed through the network. So if you do the math on this, on average, you're looking at around 1,500 searches a day, in peak times as many as 85,000, which gives you 10 searches a minute on average, in peak times as many as 12 to 1,300. So you might be thinking, 1,500 searches a day, that's not a lot. And you're right, you probably work on a system that handles more traffic. But what makes this interesting is that each individual search touches so much data, right? And this is a very niche field. So our user base is very narrow. It's essentially geneticists and bioinformaticians. So this usage is actually huge. And remember that the whole system started as an experiment. I think with these kind of numbers, we can safely say that the experiment has been validated and has been a success, and people are ready to share data. So let's go a bit deeper, and let's take a look at what kind of information people are searching for. And we can do this sort of by different parameters in the query. The first one is the reference genome. And that's actually a pretty boring one, because there's only 5 of these that the system supports. And GRCH37 is by far the most popular one. And that's actually not surprising at all. Most beacons expose GRCH37 data. It's also kind of the most common one in terms of the data that I'm dealing with on a day-to-day basis, even though it's not the latest one. So this is actually not a very surprising statistics. We find something a bit more surprising when we take a look at the second parameter, which is a chromosome. So in the chart, you see how many queries proportionally targeted a given chromosome. And you can see that out of basically 25 options, the 4 most queried chromosomes cover more than a half of all the queries, which is statistically significant. And if I had to take a guess, before doing this analysis, what the distribution would be like, I would probably expect chromosome number 1 to be in the first place, because it's the largest human chromosome. It's almost 8% of our DNA. It's very gene dense, over 3,000 genes associated with over 350 diseases. So you would expect it to be up there. So it's kind of, I mean, it's definitely up there. It's in the top 3, but it's kind of interesting to see that it's not in the first place. Chromosome 2, which claimed the first spot, actually also kind of makes sense, because it's the second largest chromosome. Again, almost 8% of our DNA, very gene dense, over 1,000 genes. So it might be interesting that it's number 1, but you would definitely expect it to be up there. What's really surprising to me is seeing chromosome 17 in the second spot, because 17 is a pretty short one. It's about 3 times shorter than the largest chromosomes. It's not very gene dense, so it's actually very interesting that this is so heavily studied. So let's take it a bit further, and let's take a look at the searches as a whole and what kind of variants they target. So in total, all our searches covered 85,000 variants, distinct variants, which might seem like a lot, but if you think about how much variation that is, that's actually not that much. It's actually a pretty focused search. So again, people study similar things. And in fact, if you take a look at the chart, the top 10 variants cover over a quarter of all the searches, which is definitely very significant. And some of them even fall into the same gene. So that's interesting. So now that we have the variants, we can take a look at some of their properties. And one interesting property to look into would be harmfulness. Basically, you want to see how many of these variants are known to be involved in diseases. And obviously, what you want to see is a high number of damaging variants, because that kind of validates the system as a discovery engine. It suggests that people are looking for stuff that they probably have seen in their own data set, and they want to see who else has seen this particular damaging mutation. So how do you determine if a variant is damaging? Well, you can do it experimentally, which is very accurate but doesn't scale to this amount, or you can do it algorithmically. There are prediction algorithms available that basically rank variants in terms of their potential for being harmful. They're not completely accurate, but it's a reasonable approximation. They typically look at different things as the basis for their ranking. So it's actually pretty good advice to run multiple algorithms if you're trying to classify something. And that's what I've done in this case. So I looked at two really common sort of classic benchmarking algorithms, SIFT and polyfen2, and I ran them across all our searches. There's actually two versions of polyfen2. There's HDiv and HVAR, which are suitable for different types of data sets. But because Beacon Network is a mixture of all the data sets, I actually ran both, and they got pretty similar results. So I'm only listing one of them here. But the interesting thing is that SIFT thinks 69% of our variants are damaging, and polyfen thinks that 55% of our variants are damaging, which is quite a high number. Even if this was like 10%, I would say that's pretty high. So 55 and 69 is definitely a very positive result. So what other properties of variants can we study? Well, one thing that was an important consideration for us when we were designing the system was its use for rare diseases. With common variants, quite often you can find additional information in various public databases. But with rare variants, finding that extra match, that's really critical, because to make new discoveries, you need to have some data to compare against. So an interesting metric for us would be how many of these variants are actually rare. So how do you determine that a variant is rare? Well, one way to do this is to basically take the variant and take a look at its allele frequency in some really well-known large data set. And that's what we've done here. So we took the 1,000 genomes data set, which is a very famous public sort of reference data set for genomic studies, and we cross-referenced all our variants and computed their allele frequencies in this data set. A rare variant is typically defined as a variant that has allele frequency less than 5%. So in our case, it actually turns out that 25% of variants could be described as rare, which again is quite a lot. Normally, you would have, let's say, 1 to 4% of rare variants in a genome, so 25% is quite high. It's a very interesting result. So OK, we took a look at variants. How can we push this even further? Well, one thing to look into would be cross-referencing variants with genes. Genes prescribe how proteins are made, so they determine traits. So that's a very interesting thing to look into. So we've done exactly that. We've cross-referenced our data set with a gene database, and we try to count the number of genes that we've hit. So in total, our searches hit almost 10,000 genes, which is actually a lot. But if you take a look at the chart, top 10 genes cover almost 50% of all the queries. So again, there's a small number of things that are very heavily studied. And I don't actually have a lot to say about these genes. I don't recognize many of them, but you can see that they're associated with things like breast cancer, pancreatic cancer, liver disease, that sort of thing. So we can take this even further, right? We have variants. We kind of extrapolate it to genes. Now we can extrapolate from genes to the actual diseases. And we can take a look at what kind of conditions people study. And that's what we've done here. So we took a look at two sources of data. There's OMIM, which is basically a catalog of gene-to-disease associations. One problem with OMIM is that it doesn't use a controlled vocabulary. So if you were doing some sort of programmatic processing, it's hard to deal with things like synonyms, which is why people are increasingly turning towards ontologies. And one really famous ontology is HPO. So HPO doesn't deal directly with diseases. It deals with clinical abnormalities, which means that you can have things that are associated with diseases. But that's interesting as well. So in terms of diseases, in the left column, you can see that people are heavily searching for cancers. There's pancreatic cancer, breast cancer, prostate cancer, and that's interesting to know. As for HPO, rows 1, 2, and 7 are not actual conditions. They're known, they're basically what's called modes of inheritance. So they're terms that describe how certain things are inherited. And of course, everything is inherited, so we do expect these terms to have many hits. As for the other stuff, you can see there are issues associated with spine, height, weight, cognitive defects, birthmarks, that sort of thing. So it's interesting to know what people around the world study. So that's all I had for you today. Everything that I talk about is open source and on GitHub. It's easy to find. Just search for GA4GH. Like I said, it's a non-profit organization, largely driven by volunteering work. So if you think that this is something you would like to be involved in, definitely feel free to reach out to me. And I can get you involved in this project or point you to right people who are involved in other projects. And there's tons of exciting stuff going on within GA4GH. If you have any questions, I'll be around for a while, so just come up and say hi. And otherwise, I hope you thought this was interesting and enjoyed the last few hours of Strange Loop.